Hello to our friends joining us via recording. Today we are wrapping up lesson number 10 about the way that neurons work. To help us remember the way that neurons work, we need to review a little bit of the content that we covered in yesterday's class. In yesterday's class, toward the end, we talked a lot about the idea of how when neurons get messages, either their membrane charge becomes more positive or their membrane charge becomes more negative. When we're thinking about a neuron, here let's do a quick sketch of Dr. Hollis's favorite graph here. When we're thinking about a neuron, remember that the only place that neurons can talk to their neighbor is up here at the very top of that graph that you have in your notes. And it looks way better in the notes, right? But so remember, when we're talking about neurons, they have to get all the way up to positive 30 millivolts. That's my charge all the way up here. For a neuron to be able to talk to its neighbor, it's got to go really positive. Neurons don't usually start very close to positive 30 at all. Neurons, when we're in normal situation down here, what's my normal charge on a neuron when it's not all excited talking to its neighbor? Yeah, exactly. When a neuron is not talking to its neighbor, a neuron lives down at negative 70 millivolts. Remember, we call that the resting membrane potential. So if a neuron is going to be able to talk to its neighbor, it's got to go all the way from negative 70 up to positive 30 that we see up here. When we want to talk to a neighbor, then, a positive change in our charge is going to get us closer to that positive 30 that we're shooting for. A negative change in our charge is going to make us go more negative or go away from that positive charge we're trying to get to. So when we think about these abbreviations, and you'll see them all over the homework assignment and all over our unit exam, when we talk about a positive change in our membrane charge, this is an example of an EPSP, because remember that E stands for excitatory. So when we have a message that a neuron receives, and that message makes its membrane charge get a little more positive, we would say that that was an excitatory message. It made us change our membrane in a positive way. We made an EPSP. If we get a message, though, and that message makes our membrane charge more negative, we would say that what happened to our membrane charge is we just made an IPSP. Remember that the I in this name means inhibitory. As in, this is going to prevent me from talking to my neighbor. If we go more negative, it is even harder for us to get up to positive 30. The more negative we go, the harder and harder it gets. So if I send you a message and that message generates an IPSP in your membrane, I'm telling you not to talk to your neighbor. I'm inhibiting you. But if I send you a message and it makes your membrane charge go up, I've generated an EPSP, an excitatory postsynaptic potential. When we talk about making these little changes in the charge of our neurons, we are talking about what happens in response to a neurotransmitter. We talked about neurotransmitters yesterday as the chemicals that neurons use to talk to each other. So if these kinds of messages are caused by chemicals that attach to a type of gated channel, not a trick question, what kind of gate are neurotransmitters going to open that makes us make these kinds of charge changes? Yeah, exactly. So when we talk about making EPSPs or IPSPs, where we change our membrane charge a little bit positive or a little bit negative, We are talking about chemically gated ion channels. Chemically gated ion channels. Remember, way back from unit number 1, chemically gated channels open with the right chemical key. Now, to help us figure out, I just use the generic term here, ion, about these channels, to help us figure out exactly which ions we're letting in or out. This is where we get to go back to my favorite, right? This is where we get to go back to that salty banana. So, lest we forget about it, here comes our banana. When I talk about my salty banana, what do I find on the outside of this banana? What do we have on the outside of a salty banana cell? Yeah, exactly. We got that sodium, which has a positive charge. We've got that chloride with a negative charge. There's my salt. And remember that just like inside a banana, inside your cells, you've got a lot of potassium, K+. When we talk about opening up a chemically gated channel for any of these things, remember we talked about this yesterday, they are always going to move from where there's a lot to where there's a little. So I've got a lot of chloride outside. When I open a channel for it, that chloride is going to rush inside. Remember yesterday we said that chloride is an anion. It's got a negative charge. So if I open up a channel for chloride, do I get more positive or more negative when I move inside? Does chloride get me more positive or more negative? Yeah, exactly. It's going to make me more negative. Hey, so check this out. We can say that an IPSP, an inhibitory postsynaptic potential, could be made by a chemically gated chloride channel. Chemically gated chloride channel. Because when my chemical attaches to this channel, chloride rushes in and brings its negative with it. When it brings its negative with it, that's an IPSP, an inhibitory postsynaptic potential. Hey, my chloride has a positive charge. Can you remind me in the chat, when we talk about something with a positive charge, what do we call those kind of ions? They're not an anion anymore. What are those kind of ions? Yeah, exactly. Sodium is an example of a cation with a positive charge. Sodium is going to come inside my cell as well. Since sodium is a cation that's coming inside, is this going to make me generate an EPSP or an IPSP? Exactly. Several of us chiming in. When I move sodium inside my cell, that's going to generate an EPSP. So I'm going to put up here on my list that chemically gated sodium channels are what I can use to generate an excitatory postsynaptic potential. If you send me a neurotransmitter, a chemical key, that opens up a channel for sodium, in comes that positive charge, and now inside, I'm more positive. Last ion that we care about, because I have chemical keys for these kind of channels too, is potassium. Potassium also has a positive charge, but potassium is going to leave my cell. If potassium leaves my cell, what's going to happen to the charge on the inside? Is it going to go up or go down? Or we could say, is it going to get positive or negative if potassium leaves? Yeah, several of us are saying it's going to get more negative, or my charge is going to drop. If I open a chemically gated potassium channel, this would also generate an IPSP, an inhibitory postsynaptic potential. Because when potassium leaves, it takes its positive with it and makes it more negative on the inside. Here's something I want to make sure that you write down in your notes, or make sure that we know for sure. When we're talking about neurotransmitters and how neurotransmitters affect the charge of the postsynaptic neuron, the neuron who's listening, we are talking about making EPSPs or IPSPs. So you see this terminology here. If I ever mention EPSP or IPSP, first thing I want you to think is neurotransmitters. The way that I make these things is when neurotransmitters attach to my channels. And the second thing I want you to always think when you see EPSP or IPSP, I want you to be thinking chemically gated channels. Chemically gated. Because it's all about those neurotransmitters. Now, I know we mentioned this a little bit in our groups, and we'll talk about it more here as a big class. When I'm talking about chemically gated ion channels that need a particular chemical to open them, in this case a neurotransmitter, which part of my neuron am I going to find chemically gated channels on? I'll take any excuse to draw a neuron. Let me draw us a neuron here. Where on a neuron do you find chemically gated channels? Yeah, Kaylin's laughing at me taking any reason to draw a neuron, right? Kira's right. When we talk about chemically gated channels, whether it's our chemically gated sodium channels, whether it's our chemically gated chloride or potassium channels, any kind of chemically gated channels, they're only found on the dendrites. So let me put 2 circles for my chloride, potassium, and sodium. I only find chemically gated channels on my dendrites. This is because, remember, the function of dendrites is to receive messages. When I receive messages, or where I receive messages, is at the dendrites. That's where I'm going to have them. Let me bounce to our actual neuron potential slide, because I want to use that. It's a little, actually I was going to say it was a little better than mine. It's a lot better than what I drew. So let's go back here. Here's that graph I tried to draw, and it looked really awful, right? When we are talking about EPSPs and IPSPs, the part of this graph that we're looking at here, the part of the graph we're talking about is right here. This region right here where it says stimulus applied. Remember that yesterday we said the stimulus we're thinking about with our neurons is neurotransmitters. So the part on this graph that's all about neurotransmitters, what kind of chemically gated channels we open up, that's right here, this little region right here. Remember, if I get to threshold, threshold is the point where I'm far enough away from normal that now my voltage gated channels take over. So once we're above threshold, you should always be thinking, these are my voltage gated channels. Anything above threshold is some kind of voltage gated channel. The only time we do chemically gated channels is right here, in this region where my neuron is trying to figure out whether or not it's actually going to reach threshold and talk. So when we get to threshold, we still need our membrane charge to go up. What was the ion, again, that helped my membrane charge to go up or get more positive when I moved it? Yeah, Kira's right. She mentioned sodium. Sodium is what I use to depolarize or make my membrane charge get more positive. If I opened up enough of those chemically gated sodium channels down here that I hit threshold, now voltage gated channels will open up, and they are all the way open. Kind of think of it like a fire hose. All the sodium rushes in all at once until we get all the way up here to positive 30. And when we get up here to positive 30, remember, my cell has an oops moment and realizes, oh, I am not supposed to be this positive. At this point, it has two goals. Goal number one is to get back to normal as fast as possible, to do what's called repolarization. Repolarization, the ion that a cell uses to get back to normal, is potassium. It's going to spit out all the potassium, all of those positives, to get back to normal. Now remember, we are not at this point on our graph anymore. We're now all the way over here, and we're dropping down. All of this stuff is voltage-gated channels. These are the channels that are going to help me get right back to the right charge because I'm at the wrong charge. So voltage-gated channels, voltage-gated potassium channels in particular, are what are going to help me get back to my normal charge. First thing I do at positive 30 is I work on getting back to negative 70, and I use those voltage-gated potassium channels to do it. Second thing I do at positive 30 is what this little symbol up here represents. I'm going to talk to my neighbor. The ability of a neuron to talk to its neighbor comes down to its ability to release neurotransmitters. So let's look at, find my picture for it really fast. Can you tell me what page this picture is on in your notes packet? Where is this picture found? Okay, I'm hearing page 11. Awesome. The ability of a neuron, here's its axon terminal, to spit out these neurotransmitters that it has inside of it, this all depends on calcium. So if you haven't already written this somewhere on your notes packet, I want you to write this down and underline, highlight, star it. The role of calcium in neuron signaling is neurotransmitter release. Let me type it for us. Role of calcium, the job that calcium plays in the process of neuron signaling, helping neuron to release neurotransmitters. The way I like to think about the job of calcium in this process of neuron signaling, think about this axon terminal here like a really cool party. There's a lot happening. We're really excited down here. An action potential gets here, meaning the membrane charge got all the way up to positive 30 down here. When it gets to positive 30 down here at the axon terminals, these voltage-gated calcium channels open up. They're like, sweet, I can finally come to the party. Except when calcium comes to the party, the neurotransmitters are like, what? Calcium is way not cool. I'm peacing out. So when calcium comes into the axon terminal, those neurotransmitters can't even. They have to leave. So voltage-gated calcium channels only open up when the membrane down here at the axon terminal gets to positive 30. When it's positive 30 down here, calcium rushes in, neurotransmitters are like, no, and they leave the axon terminal. So the only role of calcium, it doesn't change the membrane charge. We don't bring in enough of it to make the membrane more positive. We just bring in enough of it so that those neurotransmitters leave. When we're talking about the kinds of ion channels that we find down here at the axon terminal, one of them that we find down here is the voltage-gated calcium channels. voltage-gated calcium channels. This is the ones that we just talked about. So the job of the voltage-gated calcium channels, that helps us to release those neurotransmitters. But remember, I told you that these channels only open up when the membrane down here gets to positive 30. Does anyone happen to remember what was the kind of ion channels that helped a neuron get to positive 30? What's the one that helps us to get there really fast? So after threshold, who depolarizes us? Yeah, it's those sodium channels, right? And specifically, it's those voltage-gated sodium channels. Oops, voltage-gated sodium channels. These are also found at the axon terminal. Voltage-gated sodium channels, their job is to depolarize or to make positive the membrane down here. So I've got voltage-gated sodium channels that they're not showing me down here, but I've got them. I know that I've got them because these voltage-gated calcium channels opened up, and in came that calcium so that these neurotransmitters get kicked out. But if I'm all the way up at positive 30, I do not want to stay there forever. The other kind of channel that I find down here at the axon terminal is the kind of channel that's going to help me get back to normal. The kind of channel that helps me get back to normal after I've completely depolarized are those voltage-gated potassium channels. I spit out potassium to get me back to normal. These are the ones that repolarize the membrane, or they make it negative. They make it negative. You have questions on the homework assignment that ask you about the kinds of channels that we find at the axon terminal down here. There are 3 kinds that we find at the axon terminal. The first kind, we only find it there. It's nowhere else on the neuron, just at the axon terminal, those voltage-gated calcium channels. But the other 2 kinds that I find at the axon terminal are the voltage-gated sodium channels that I use to get to positive 30, and the voltage-gated potassium channels that I use to get back to normal. So make sure when you're working on that assignment and it asks you about the axon terminal, I'll add that up here. We're talking about the axon terminal right here. Make sure you tell me all 3 of these types of channels. Hey, study pro tip. I'm going to go over here to the other side. Check this out. You know how Dr. Aulis is tricky, right? All too well. I promise this is not a trick question. This one's not. Have we talked about chemically gated calcium channels? Have we talked about that? I'm not trying to be tricky. Yeah, Kira votes no. Kira's right. We have not talked about chemically gated calcium channels because there are not chemically gated calcium channels. Please read slowly. Chemically gated calcium channels don't exist. I don't use them because all I use chemically gated channels to do is to hear messages from my neighbor. And remember, the only thing that calcium is supposed to do is help me to spit out neurotransmitters at the axon terminal. All of my chemically gated channels are found on my dendrites, but I do not have chemically gated calcium channels because my dendrites don't ever spit out neurotransmitters. Only the axon terminal does. So please, please, on the homework assignment, read the names of the channels carefully. And please, please, on the exam, read the questions carefully. This kind of channel, chemically gated calcium channels, does not exist. We do not have these on neurons. we've got voltage-gated calcium channels, and we've got chemically gated all kinds of channels. Chemically gated sodium, potassium, chloride, just not chemically gated calcium channels. Yeah, I like it. Like Tien said, this is fake news, not real. Don't tell me that this is the kind of channels that we have anywhere, because they don't exist. I'll mention, we'll go back and talk some more about channels, but I want to mention these two things you can see on the picture down here. When a neuron spits out neurotransmitters, so when those neurotransmitters couldn't even, because calcium came in, they go out into this space that I think we talked about in lab briefly, called the synaptic cleft. So neurons don't actually touch each other. The neurotransmitters that one reaches out or spits out floats across this synaptic cleft and attaches to those chemically gated channels that we started with yesterday when we were talking about our EPSPs and IPSPs. So neurotransmitters go across the synaptic cleft. They attach to these chemically gated channels. Once a neuron responds to the neurotransmitter, two things happen. First thing that happens is we respond to it. So we're going to make that EPSP or that IPSP. Second thing that's going to happen is we're going to find a way to get rid of that message. Remember, like we were talking about with sarin gas, how it can be a really big issue if we hear the same message over and over and over again. So there's a couple of ways that neurons address this to make sure they don't hear those messages over and over again. One of them is using an enzyme to break down that message. So that's the exact same enzyme that we talked about with muscles. Remember that enzyme, acetylcholine esterase. This is one example of an enzyme that we could use to break down our neurotransmitters. We also use other different enzymes to break them down. This one only breaks down acetylcholine. When neurons talk to each other, they can speak more than one message. So if the message we're speaking is acetylcholine, sure, we can use that enzyme. If it's a different enzyme, we're a different neurotransmitter, we'd use a different enzyme for that as well. So one thing I can do when I'm done hearing a message is I can chop up that message so I don't hear it again. Another thing I could do if I am feeling a little bit thrifty, a little bit savvy, is when that message detaches from the chemically gated ion channel, I might take it back up. Save myself a little energy. Don't make a new neurotransmitter if I don't have to. So sometimes we take those neurotransmitters back in. Sometimes those neurotransmitters just kind of diffuse away. They float away. This is not necessarily our primary mode of making sure that we don't hear a message repeatedly, but this is another option we have as well. So we can chop things up, they can float away, or we can do what's called reuptake. Now check this out. Perhaps you have heard of a kind of medication called a selective serotonin reuptake inhibitor. Selective serotonin reuptake inhibitor, or if we abbreviate it, an SSRI. SSRIs, or selective serotonin reuptake inhibitors, this is a kind of antidepressant. Can we predict from its name how might an SSRI work? What do you think it might do? Name. Yeah, so the most basic, based on its name, what we could say is it's going to block us from reuptaking or stealing this serotonin back into the other cell. So that means that normally when I would have heard this message once, I didn't take it back inside, so maybe I'll hear it twice or 3 times or 4 times. This is going to allow me to piggyback with what Audrey's talking about, This is going to allow me to generate signals like EPSPs more than once from just one neurotransmitter. See, in depression, sometimes an individual is not releasing enough serotonin. Any serotonin that their neuron spits out, they want to hear it as many times as possible. So a selective serotonin reuptake inhibitor allows that message to be heard more than once. It's going to block this pathway right here. So when my neuron spits out 5 serotonin molecules, maybe it's going to be heard more like 10 or more like 15. And serotonin, again, a neurotransmitter that's not released enough when an individual is depressed. So this helps them to hear those happy messages, if you will, more times. A selective serotonin reuptake inhibitor. Any questions about the axon terminal or about our channels, about SSRIs? I also need some emojis because we have not done nearly enough emojis today. Here comes some emojis from Dr. Hollis. I like it. Got a dolphin. Kaylin, when I see the upside down smiley face, I think you're feeling kind of whimsical. That's probably overreading it, but we'll say that. Audrey's dreaming of the beach. Yep, I feel ya. Nice, Tiana, I like that. Investigating. Perfect. Okay, let me look back at our pictures here. Let's see where to go next. Let's talk about a quick refresh here. If this picture looks familiar to you, it's because it's literally the same picture we saw in the muscles chapter. Remember in the muscles chapter when we talked about how a muscle gets a signal to contract at the neuromuscular junction, so right there at that spot, the membrane charge changes. Then a muscle cell uses those T tubules to send that message everywhere inside the cell. When I'm talking about a neuron, hey look, I've got an excuse to draw another neuron. Here we go. When I'm talking about a neuron, I don't have those T tubules to worry about so much, but I am receiving my messages up here at those dendrites. And for me to have any effect, for me to talk to my neighbor, that's got to happen all the way down here. So when we think about these action potentials, which again is what it's showing you right here on this picture. In a muscle cell, the goal of an action potential was to get the whole muscle to contract. When we're talking about a neuron, the goal of an action potential is actually to get my message from point A, where we received it, all the way down to point B, where we're passing it along. The same ideas apply with these channels when we talked about them in muscles, just like when we're talking about them here in neurons. Does anyone remember what we call this little triangle area right here? No, it's not technically a triangle, but I'm not good at drawing. Yeah, exactly. Okay. This is my part of the neuron called the Axon Helic. The Axon Helic is where I do summation. That was the last thing we talked about in class yesterday, remember? How summation is when I mix together all of the messages that I'm hearing. Some of them are positive, some of them are negative. That helps me to know whether or not I'm going to hit threshold. The axon helic up here is the first part of a neuron where we start to see voltage-gated channels. Voltage-gated channels. So starting up here at the Axon Helic, where we're adding together all of our positives and negatives, if we hit threshold right here, we're going to start opening up the voltage-gated channels that would allow me to send a message down my neuron. So imagine that this top part of my picture up here, that's right here. So first I open up voltage-gated sodium channels, and that makes this part of the axon positive. Well, if this part gets positive, the voltage-gated sodium channels that live right here, they're going to open up too, because it's positive, which makes these voltage-gated sodium channels open up, which makes these ones. And action potential is when we called it before the domino effect. When these voltage-gated sodium channels open, all of the rest of them are going to open all the way down to the axon terminal. But when these ones open, the voltage-gated sodium channels, remember that I also have to open up the voltage-gated potassium channels after them. Because when these channels are done, when the voltage-gated sodium channels have done their bit and they've finished up, we're at positive 30. And this part of my neuron does not want to stay at positive 30. It's not supposed to be there. So then those voltage-gated potassium channels open up, and we go back down to normal. And then these voltage-gated potassium channels are opening, and these voltage-gated potassium. So all up and down my axon, all the way down to the axon terminal, that's where I find the voltage-gated channels. Everywhere where I'm sending a message, from where I first hit threshold here at the Axon Helic all the way down, I'm going to have voltage-gated potassium, voltage-gated sodium channels, because they help me to move my electrical signal. They give me an action potential. So you have, let me pull up the picture. You have this picture in your notes. What page is this picture on here? This picture here. We're all scrambling. Okay, awesome. We're on page 12 now. I know I'm bouncing you around everywhere. I apologize. We're on page 12. We just did this. Yeah, Kaylin says she drew this like 6 times. Hey, that's awesome. You will know your neuron parts and your channels. Look at our options here. We want to know where on our neuron we've got chemically gated chloride channels, chemically gated sodium channels, chemically gated potassium channels. Remind me in the chat, when we've got chemically gated channels, what do they open with? In particular in my neurons. What do chemically gated channels with my neurons, how do these guys get opened? Yeah, exactly. That was a mean one for me to ask you. That was a long word to type, right? Neurotransmitters. Neurotransmitters. When I'm talking about opening chemically gated channels, I only find chemically gated channels in places where there's going to be chemicals. So the only places that I'm going to have chemicals are going to be those dendrites, where I'm receiving information. So my chemically gated channels, I'm going to do a really bad job at kind of tracing around all my dendrites up here. Chemically gated channels all over those dendrites, because that's where I receive chemicals. Never, ever, ever tell me there's chemically gated channels down here. No chemicals involved down here at my axon terminal. Never tell me there's chemically gated channels here. We do not have chemically gated channels here. Because see, from the Axon Helic down, we are all about sending messages. When we transition over from the receiving messages to the sending messages, which is what's happening here, now we're into the realm of voltage-gated channels. Voltage-gated sodium, voltage-gated potassium, voltage-gated calcium. Hey, one of these 3 types of channels, one of these 3 I do not find along the axon. Who does not live along the axon? Pick my color here. Yeah, Kira's right. It's those calcium channels, the voltage-gated calcium channels. Remember, the one goal in life of the voltage-gated calcium channels is to get neuron to spit out neurotransmitters. That's the one goal in life of voltage-gated calcium channels. Get that neuron to spit out its neurotransmitters. So the only place on my neuron where I'm spitting out neurotransmitters is down here at these axon terminals. So the only places you're going to find voltage-gated calcium channels are down here at the axon terminals, the places where we spit out our neurotransmitters. But anywhere where the membrane charge changes, because remember, that's the job of the voltage-gated potassium and voltage-gated sodium channels. Anywhere where we're sending a message for the membrane charge to change, so all along the axon and all along my axon terminals, I'm going to have voltage-gated sodium channels and voltage-gated potassium channels, because they're going to allow me to take the response, or take what I got from summation. I do summation here, add those EPSPs and IPSPs. Voltage-gated channels allow me to take that message all the way down to where I can do something about it and pass it along. What questions do we have about the locations of channels on a neuron? Or shoot me a thumbs up. Uh-oh. Expound, Kaylin. You can't just ugh me without an expounding. Audrey likes the picture. Or you're looking for something that you'll remember this by. I can't tell which way you were. Is that a comment or a question, Audrey? Oh yeah, I like that. That's a great way. We had done that before, and I didn't even think of that. That's really true. She likes to remember the chemically gated channels around on the dendrites as a C around that cell body. I love that. Here I'm trying to color in a C really fast, and it's looking really awful. But I like that. Chemically gated channels up here, big letter C around the cell body. I love that. That's perfect. That is a great study tip. I'm going to steal that, Audrey, when I teach this class next semester. I'm totally gonna steal that. So thank you. That's great. Chemically gated channels up there. I like it. In addition to knowing where those channels are on the neuron, the other thing that I asked you to do was to know when on this graph we're using these different channels to do things. So when we're looking at this graph, remember and you have another label where you're labeling the parts of the graph. Well, here, let's go back and we'll do that really fast. I'm going to put numbers, and I'll have you help me out in the chat. So 1, 2, 3, 4, 5, 6. My numbers are really terrible. I apologize. When I am talking about the part of the graph where we're doing well, no, let's just go in, we'll go in number order, okay? When I am over here at time number 1, what do I call time number 1? When I'm down here at -70, normal, if you will. Yeah, exactly. That's what we would call resting membrane potential, number 1. That's where we chill out. Remember, we do have to know that number. We do need to know -70. That's an important number. -55, my other important number That's number 2 on my graph. What is point number 2 representing? What do we call that negative 55? Yeah, exactly. We call that threshold. Threshold is time point number 2. Absolutely. If we hit threshold, remember, this is when the light switch goes on. And now we are going to completely change our membrane charge. Yeah, now we're going to do, just like Kaylin says, now we're going into depolarization. Number 3 is depolarization. I like that. Audrey says, think about hitting threshold as we open the door and we say, come on in. Yeah, we tell sodium, come on in. It's great inside here. And we get completely depolarized. We go all the way up to positive 30 before we kind of have an existential crisis and realize we should not be positive 30. We're going to talk to our neighbors, right? So point number 4 on this graph is when we spit out a neurotransmitter, because neurotransmitters are what we use to talk to our neighbors. So at point number 4, we're doing neurotransmitter release. Only if we're at the axon terminal, right? That's where I have those voltage-gated calcium channels to help me bring in the calcium so that the neurotransmitters will peace out and disappear. The other thing I do, though, at positive 30 is some soul searching and realize I need to get back to normal. What's the word that I use for I'm trying to get back to normal? Yeah, exactly. Like Kaylin and Kira mentioned, repolarization. That's the word I use for I'm trying to get back to normal. That was a really terrible number 5. That's a little bit better. Time number 5, repolarization. I'm trying to get back to normal. Here's the thing, though, with this trying to get back to normal. The problem is my channels that I use to get back to normal, they're the voltage-gated potassium channels. They're so excited about spitting out potassium that they spit out too much, and I actually end up too negative. I spit out too much potassium, and I get really negative. Yeah, and what I call it when I'm down here and I'm extra negative, that's the part called hyperpolarization. Oops. Hyperpolarization, when I'm too negative or too much polarized. Eventually, I'll fix it and I'll get back to normal. But down here at the bottom, when I'm too negative, I would be what's called hyperpolarized. Yeah, Caitlin mentioned in the chat, this graph is on your homework assignment, and you can do it as many times as you want, or maybe more accurately, as many times as you can stand, right, until you get it right. So here's labeling the parts of the graph. Now that we've given them their names, now we should pair that information with the kinds of channels that are involved. When I'm talking about this part of my graph right here, this is the part of the graph, remember, where I'm doing summation. This is the part of the graph where I'm doing those EPSPs and those IPSPs. We talk about making EPSPs and IPSPs. Which kind of channels were we using to make those kinds of changes? Yeah, exactly. This part of my graph is where I'm going to use those chemically gated channels, my chemically gated channels. Technically, the kind of channel we're seeing here, like Kira mentioned, if we just have excitatory messages being made, then that's going to be all about sodium. Sodium is my only option to get us positive. But if before I had all these EPSPs, had some negative stuff going on too, it would be my other kinds of chemically gated channels as well. Big emphasis being the only time on this graph where you've got chemically gated channels involved, any of my chemically gateds. So sodium, potassium, chloride. It's all right here at this point in the graph. Because remember, when we get here to threshold, when we hit threshold, we are opening up the door and we're saying, you can come on in. Which kind of channel would we say is opening the door to let things inside? Who opens up right here at threshold? Who opens here? Yeah, so when we talk about what's going to help us get positive and shoot that membrane charge up, this is the job of voltage-gated sodium channels. Voltage-gated sodium channels. They first open up at threshold. -55 is the voltage that opens them up. They open here, so when here's a wink, wink, nudge, nudge. On the homework assignment, when it asks you about which kind of channels first opens at threshold, voltage-gated sodium channels first open there. When it asks you about the kind of channels that we use to rapidly depolarize the membrane, it's still these guys. It's still my voltage-gated sodium channels. We labeled them on here twice to remind ourselves that here's the point that they open, and here's what they stay open through. They're going to close up here at the top. When we get all the way up to +30, these channels are like, oh, right. I'm not supposed to be making us any more positive. So voltage-gated sodium channels will actually close up here at the very top. When we're up here at +30, there's two kinds of channels that can actually open up here. Let's start first with the kind of channel that opens to help me send a message. Which of my kinds of channels helped me to send a message? Yeah, the kind of channel that its only job in life, remember, was to help me send messages, was the voltage-gated calcium channels. Voltage-gated calcium channels. These were only seen at the axon terminal, at the end of the axon. They opened up, calcium rushed inside, and remember, those neurotransmitters couldn't even. They just had to leave. I can't even. That's how we talk to our neighbor. In addition to opening up voltage-gated calcium channels, we also open up the kind of channel that helps me to repolarize. Who helps me to repolarize? Who gets me back to normal? Yeah, exactly, Temiteo. I like your abbreviation. I usually do that. I hadn't so far today. We're doing it now. Voltage-gated potassium. Voltage-gated potassium. That's what I use to repolarize the membrane. If your question asks you about when they open up, when voltage-gated potassium channels open up, they open up here at +30. Just like voltage-gated calcium channels open up at +30, these ones open up then too. Except the entire axon is covered in voltage-gated potassium channels. Only the axon terminals have voltage-gated calcium channels, because that's the only place we spit out those neurotransmitters. Voltage-gated potassium channels, on their way back down. We're getting the membrane charge back to normal, back to normal. I'm really bad at closing, so we get a little too negative. At this point, we switch over to using those leakage channels. What can you remember about leakage channels? Can you tell me what we remember about those? Yeah, exactly. So leakage channels are a lot like that dripping faucet, right? They move stuff really slowly, but they're always moving stuff. Part of what my neuron uses to get back to normal is some leakage channels that helps it to stabilize back more toward normal. The other thing it'll use to help it get back to normal, I didn't make you label it on here, so don't worry too much about this, but the other thing we use to get back to normal is the sodium-potassium pump. That thing we talked about way back in unit number 1 that I told you polarized the membrane. If we get hyperpolarized, this is going to help us pump stuff back the right direction, get it back to the correct charge, back to resting membrane potential. I want to pause for a moment and give you a chance to think of any questions you might have. Can I get a thumbs up? Few thumbs up. Good. And what I'll mention for you as you look at this graph, it may completely blow your mind still. I 100% recommend this graph, the graph that we filled in before with the different parts, so depolarization, repolarization, all that jazz. All this stuff on that page of the packet, from the neuron through these graphs. Print this stuff out like 5 times and do it like 5 times. Because I promise you, you will see these graphs on the exam. Like, I'm not even going to pull any punches. I'm telling you, you're going to see the graphs. You're going to see questions about the neuron, where we find those channels. So I promise you it's going to benefit your studying to do those activities over and over again. Kaylin approves of it, right? She said that she had already done it like 6 times. So take it from Kaylin that it helps to draw those neurons, label those channels. Same thing with the graph. Make sure we catch those things. I want to transition into talking about something called the refractory period. So let me find my best slide for this. Let's talk about a time called the refractory period. And can you help me out in the chat? What page are we on now? Okay, we're on page 15. Excellent. Okay. When we talk about the refractory period for a neuron, this is the period of time where a neuron either cannot respond to a message at all, like there's no chance at all that it can, or we have to hear a message really, really loudly to be able to respond to it. When we talk about the refractory period, there's the first part where there is absolutely no chance that a neuron's going to hear a new message. That's called the absolute refractory period. Something I want you to make sure that you write down in your notes about the absolute refractory period. The absolute refractory period is an issue with our voltage-gated sodium channels. The absolute refractory period, this is a problem with the voltage-gated sodium channels. Voltage-gated sodium channels, their job was to depolarize the membrane. Their job was to get a neuron all the way up to positive 30. For as long as my voltage-gated sodium channels are already open, so for as long as they're responding to another message that they just heard, they're not going to be able to get a new message. That's sensory overload. They're in the middle of getting ready to talk to their neighbor. They can't change their message halfway through. Voltage-gated sodium channels, when they're still open in response to a message they heard before, it doesn't matter how loud you scream your message at a neuron, they can't process it. They can't hear it because they're already open. They're already freaking out. Let's say, though, we've hit this positive 30, so we've hit the point where a cell realizes, oh, I am too positive. I should not continue to bring in sodium ions. I need to get negative again. One of the ways that a neuron addresses that is by having a funky shape to its voltage-gated sodium channels. So I apologize, I should have started with this picture. When we are going up our graph and we're depolarizing a neuron's membrane, sodium is rushing inside. We need this channel to be open because of the membrane charge, and we need this little plug, if you will, to be out of the way. When we get all the way up to positive 30, it's so positive on the inside that this part of my channel protein actually pops into my channel and blocks it. So I like to think about this a lot like a stopper on a bathtub. If you put this in the drain, nothing's going to go down that drain. When we get too positive in the neuron, when we get up here to positive 30, this part of my protein actually folds inside and blocks the channel. So at this point in the graph, all the way until we get about back to threshold, kind of back around the threshold value, we're going to have this blocking protein or an inactivation protein blocking the channel on our protein. It's not until we get all the way back to below threshold and resting membrane potential that our protein is restored to its normal shape and it's ready to hear messages again. So for you to be able to hear a message, your voltage-gated sodium channel needs to look like this. When we've heard a message, it changes its shape and opens up the channel so that sodium can rush inside. When too much sodium rushes inside, the inactivation part of this protein pops into place. And until the inside of the cell is back to normal, it's going to stay in this shape. When we get back to normal, we look like this. And when we look like this, now we can respond to a new message. This part shown here in green, this is my absolute refractory period. The absolute refractory period actually includes two parts on this graph here. The first part of the absolute refractory period is when the voltage-gated sodium channels are already open. I can't open them again if they're already open. So if you try to talk to me in a way that's supposed to open my channels, sorry, I can't hear you. I'm already yelling at somebody else. I can't hear you. If you try to talk to me in a way that should open up the channel, except I'm too positive on the inside and I've got my stopper in place, I still can't hear you. The absolute refractory period, the period of time when the voltage-gated sodium channels are in the wrong shape, either because they're open or because they're blocked. As we can see on this side in the green, where we don't have, so notice the upswing on my graph, that's when those channels are open. That's my picture right here. From this point, the very top, to down to where we stop being green, that's when we got this going on, when we're inactivated. That's my absolute refractory period. I absolutely cannot respond to a message that you sent me because either my responding protein is already busy or my responding protein is broken. No messages, absolutely none. We also have this time period, though, after a neuron got a message called the relative refractory period. The relative refractory period is when I could hear a message, but it has to be really, really loud. Think about the relative refractory period kind of like you're skydiving and someone's trying to talk to you. The charge on the membrane of my neuron is dropping really, really, really fast. But if I'm sending a message, that message is supposed to make my membrane charge go up. We're diving down really quickly. My membrane charge is dropping like crazy because of my potassium channels. It's going to take a really loud message, or it's going to take a whole lot of sodium to make me be able to generate a positive charge to get me back to threshold. Because see, here's a big idea with neurons. For a neuron to hear a message, It has to get to threshold. For a neuron to hear a message in the way that we normally think about this, the neuron has to get to threshold. Right now, in the absolute refractory period, this neuron is nowhere near threshold. The channels that would get it to threshold are already busy. So it's not going to get to threshold in response to the message you just sent because it's already on its way there. When we're on this side of the graph, when those voltage-gated sodium channels are available, we are dropping down fast. We're skydiving, and you're trying to send me a message that's supposed to make me get up to threshold. Yeah, good luck with that, because I am plunging to the ground. It's not until we get back to around our normal or our resting membrane potential that we leave this relative refractory period. The reason we have the relative refractory period is because of those voltage-gated potassium channels. Voltage-gated potassium channels are trying to make our charge get more negative. When they're open, you're skydiving. You're going to have a really hard time going back up. When we're in the absolute refractory period, you just can't hear anything because you're too busy yelling. Absolute refractory period, that's an issue with the sodium channels. Relative refractory period is an issue with the potassium channels. I have talked way too much. Tell me what we think or what we do or don't understand about the refractory period. Yeah, Kaylin mentioned she's going to go back over it. This is one of the more challenging topics of this lesson, so that's totally fine. I spent a lot of time on this topic with students last semester. Yeah, your brain hurts, your neurons hurt, right? We're somewhere in one of our refractory periods, right? Either the absolute or the relative. So much information, can't hold more, right? Hey, so Dr. Ollis is a nerd and likes the occasional meme. Here's a meme for any Lord of the Rings fans. So for anyone who is not a Lord of the Rings fan, and to be fair, I'm not really a big fan either, but my husband is, there's a phrase in that movie where it says, one does not simply walk into Mordor. That's like the terrible place that they have to go return the one ring of power. So when we're talking about our neurons, one does not simply send a new message or conduct an action potential when we're in the absolute refractory period. You cannot do it simply. It's very hard. So the refractory period, not great for sending other messages. Let's talk about, as we kind of wrap up this chapter, let's talk about the importance of myelin. The importance of myelin to our neurons and to the nervous system. Remind me in the chat, what are some of the things that we know about myelin? Okay, yeah, so myelin is made out of kind of a fatty substance, absolutely. Yeah, it's a protective covering for our neurons. I kind of like to think about like myelin as insulation, if you will. So we've got this fatty insulation layer. Yeah, it's made by either those things from lab we talked about called the Schwann cells, or those cells called oligodendrocytes. The goal of myelin wrapping around these neurons is twofold. First goal is to help us send messages quickly. So we'll look at in a moment how messages, I mentioned this before, they kind of jump between the spaces in the myelin. So one of the goals of myelin is to make messages get sent quickly. The other goal of myelin, though, is to help messages get sent correctly. If this neuron is supposed to send a message to its neighbor that's a really strong message to fire, so we're supposed to go talk to 10 friends, we want to make sure that that message gets down and gets communicated correctly by the end of this neuron. If my myelin sheath becomes damaged, or if we mess with it, we might send the incorrect message, and it may be that by the time this signal gets to the axon terminals, there's not enough of it to even to our neighbors. So myelin makes sure that messages go quickly and that they go accurately for us. So here's how the process works when we're talking about a neuron sending a message, either using myelin or not using myelin. At the beginning of the axon, we open up voltage-gated sodium channels. Voltage-gated sodium channels rush in a bunch of sodium and make this big positive charge. So we can see the big positive charge right here. Now, that voltage-gated sodium channel with its big positive charge, it brings in the positives, and they start to work their way down the axon. Notice how the size of this charge, it started really big, and really quickly, it's getting a lot weaker. So when I don't have myelin, I have to put in another voltage-gated sodium channel, not that far from my last one, to bring in a bunch of new sodium, because as I move a little ways, I'm still losing my charge. I got to put in another one, and another one, and another one. When we talk about a neuron sending a message without myelin, there are voltage-gated sodium channels all along the axon. We need to open up those channels repeatedly. Yeah, Audrey has an analogy that works great for me. When we've got a non-myelinated neuron, these are all the cups of coffee it's going to take to make it through the day. Absolutely. They got to stop and recharge like every 2 hours or less. When we talk about having a myelin sheath, in comes my charge, or my sodium, bringing the inside positive. Notice that it takes a lot longer for the strength of my signal to go down, which allows me to focus or just put my groups of voltage-gated sodium channels together in those gaps, those things in lab that we called nodes of Ranvier. Nodes of Ranvier. The only place on the membrane of a myelinated axon that we actually put these voltage-gated sodium channels is inside the nodes of Ranvier. So all of them open at once and rebuild that big signal. That signal is bigger and stronger, so it lasts longer, and we can make it farther down the road. So the myelin sheath is an insulator. It makes sure that none of my electricity disappears to the outside. It also makes it so that this place here in the gap in the nodes of Ranvier, this is where all my charged things like to hang out. So all of my sodiums, all of my potassiums, they're going to float around right here because it's almost like they can smell or detect that here's where charge is. When you've got fatty substances on other places, you can't sense the charge. When you're wearing a big winter coat, the parts of your body that are covered in the coat don't feel the cold outside. It's just that little tiny space, right, between your gloves and your coat. You can feel the cold there. That's the same way it is with these myelinated neurons. We can only change the electricity just at those little gaps. Otherwise, that electricity stays strong and we're in good shape. Yeah, Audrey asked an important question. She asked about whether or not all neurons have a myelin sheath. The answer to that question is no, not all neurons do. And as we move into lesson number 11 about the brain, you will see that the brain has white matter and gray matter. And that has to do with the amount of myelin it has. Let's see if we can take a guess. Myelin is kind of fatty. If I'm putting some fat around my neurons, do you think that's going to make them look lighter in color or darker in color if I put some white or some fat around them? Some adipose tissue. Yeah, so this myelin fatty layer makes those parts of the brain look like what we call white matter. We have a lot of myelin, a lot of fat. That makes the tissue look lighter in appearance. The types of neurons that do not have myelin sheaths, those are going to be darker in appearance. They'll be what we call gray matter. Yeah, and it does, like Kaylin mentioned, it does kind of relate back to the white fat and brown fat article. Absolutely. Yeah, we got some kind of white fat going on, if you will, where there's a whole lot of lipids. And then remember from our brown fat article that they're brown because they've got mitochondria in them. So in this case, we're gray just because we don't have that white fat that we have wrapped around us. So big picture, myelin really good for sending messages quickly and sending messages correctly. By having this myelin sheath, we actually are able to skip some parts of the membrane. So this is that picture we looked at before. Imagine with me, so here I'm gonna pretend here. Imagine with me that I have a section right here that I have covered in myelin. If I cover this section in myelin, the next place that I'm going to open up those sodium channels is right here. So I'd actually be able to skip opening up the channel somewhere, and I do this all along the membrane. So I would make areas with myelin, and it helps me to skip having to depolarize the membrane and repolarize it, because there's some places that I just don't have to do that on. So this is what we call, let me bounce back here, when we're talking about having a myelin sheath, this is what we call saltatory conduction. Saltatory conduction. When we talk about not having a myelin sheath, where the entire membrane has to change its charge, that's called continuous conduction. Continuous conduction, where we have to change the membrane charge every single place. Hey, not a trick question. Do we think that continuous conduction or saltatory conduction is going to be faster? Which one do we think might be faster? Continuous or saltatory? Yeah, exactly. Saltatory conduction is much faster. The other analogy I kind of like to give with this too is think about if you were running down a hallway or like you were in a relay race, and every time that we see right here, there's a door that you have to open and go through. If we're doing continuous conduction, like every 5 feet, you have to open a new door to keep going. When we're doing saltatory conduction, I can dash and then open them all at once and dash and open them all at once and dash. So whatever analogy works best for you, keep in mind that continuous conduction, a neuron has to work a lot harder and it goes a lot slower compared to saltatory conduction, where we don't have to work quite as hard and that message goes a lot more quickly. That's all I've got for today. I want to open the floor for questions or any topics that we want to discuss. We've got a little bit of time left today. Anything in particular we want to chat about? I agree. I'm very happy it's Friday. Get you your daily penguin while we think. He's happy it's Friday too. Yeah, so the question is, what is actually the dendrites? Yeah, the little tentacles themselves are the dendrites. So lining the tentacles, so here's why we kind of circled them before, is if we drew little lines on here to represent those chemically gated channels, the little chemically gated channels are all over the outside of those little tentacles, the little dendrites. So yeah, the dendrites themselves are the tentacles. We cover those tentacles with the chemically gated channels. Yeah, you're welcome. All right, well, if there are no other specific questions, I will go ahead and stop the recording. I will chill out for a few more minutes for any last minute questions you think of. Otherwise, thinking about our schedule for next week, we will definitely have class Wednesday, Thursday, and Friday because Friday will be an exam review. I need to look over the amount of content we have to determine if we need a Tuesday class too or not. So be watching for that. I'll definitely post it on the calendar so you know when we have class. But for sure, I will see you next week, Wednesday, Thursday, and Friday. Good luck with your studying.